So this is lecture 18, and just a couple of notes. Don't forget, this Friday we have our second midterm. The format is the same as the first midterm. 30 multiple choice questions. You'll have 50 minutes to complete. You can start the exam anytime between 8:30 a.m. and 8:30 p.m. So we don't have any class on Friday. If you're looking for a place to write the exam, you can utilize this room because nobody will be in here. And if you have any questions, I'm going to have extra office hours tomorrow from 11 until noon. Outside of that, you can email me and I'll see if I may be able to answer your questions that way. Again, save the date. December 20th is the tentative date for the finals. Again, this is just the preliminary schedule, but in my experience, the preliminary schedule almost never changes. All right. We've been looking at the ER Golgi lysosomal system. And we talked about it, and we looked at how components move between these systems in these vesicles. And the important thing is, once something enters here, if we look at these little blue dots, they can go all the way through this system and eventually be secreted without ever coming into contact with the cytosol. And that's important because we know the chemical composition of these compartments is more similar to the external environment than the cytosol. And we know that the way a protein folds in one environment will be different in another. So if we want these to be functional outside of the cell, we fold them in an environment that's similar to the cell. The top of this slide is the ER. And we know that we get folding of proteins in the ER and different modifications, including addition of that block of N-linked glycosyl groups. In the Golgi, as we move from cis to trans network, what happens is there's a whole host of other modifications that occur. The N-linked sugars may be further modified, things may be added or removed from that, but adding new N-linked sugars does not occur outside of the ER. O-linked glycosylation, however, can occur in the Golgi. So that's addition to carbohydrate or carbohydrates to the hydroxyl group of a serine or a threonine. The lysosomes are important for breaking down macromolecules, and they do this under acidic conditions. So they have a cohort of acid hydrolases. These are proteins that can hydrolyze macromolecules. And hydrolysis, remember, is when we add a water to a covalent bond, breaking the bond. And then finally, what we had been examining was we've been looking at the mitochondria. The mitochondria has two membranes, the outer and the inner membrane. And so that creates kind of three compartments. We have the cytosol, the matrix, it's labeled in here, yeah, is in the inside, and in between we have this intermembrane space. Right? And the intermembrane space is where we end up pumping our protons during the electron transport chain. So we very briefly looked at a couple of metabolic processes, and again, we're not doing these in detail in this course this year. You will get a lab on this, and we've given you enough details to get through that, but we'll look at this in a lot of detail in Biochem 299, or if you take Biochem 300B. So we'll go more into the energetics of this. But importantly, if we're looking at humans, we start with an organic fuel source, and what we do is we oxidize it. We pull electrons out of it, and in that process, we generate ATP. If we're looking at the breakdown of glucose, the initial step is called glycolysis. And glycolysis really, we take a 6-carbon sugar, we produce 2 3-carbon molecules called pyruvate, we produce a couple of ATP, and we harvest a few electrons in these electron carriers. NAD plus when they're empty, NADH when they're full. Pyruvate then moves into the mitochondria, which is why we were interested in this. And it's modified to this compound called acetyl-CoA, and this is carried out by something called the pyruvate dehydrogenase complex. We can just call that PDH. So this first step, we get our first fully oxidized carbon dioxide, and again, we got that through an oxidation of pyruvate, meaning we stripped electrons out, and that filled up a carrier. These two carbons are attached to what's called a coenzyme, and this enters into our citric acid cycle, so said because we end up exactly where we started. And during the process, we can see two carbons come in, shown in gray, and two carbons that are fully oxidized come out. And we fill up a whole bunch of our electron carriers that we equated to kind of little dump trucks moving electrons around in the cell. Of course, now we've filled up our empty carriers, so our NAD plus and our FAD, and what that means is if there's no more empty carriers, all of these processes, glycolysis, pyruvate dehydrogenase complex, and the TCA cycle will all grind to a halt because there's nowhere to put those electrons. And so those need to be regenerated, and one of the major ways we do that is through what's called an electron transport chain. And we equated this electron transport chain to our electron dump truck being at the top of a hill, dropping the electrons, and as they roll down the hill, they move a pump, and those pumps move protons across the inner membrane of the mitochondria into the intermembrane space, which is an example of active transport. And we're going to pick it up with that today. Any questions? No? All right. So again, what we're looking at here is kind of a close-up. You don't need to memorize this system. But what we can see here is our NADH dumps its electrons into this, which we call complex 1. And the electrons move, if we follow these red arrows, we can see that they move, and eventually, as we talked about in the last class, those electrons have to go somewhere, and where they go is oxygen. And so at the end of this whole thing, electrons go onto oxygen. When we reduce the oxygen, we reduce it to water. And so we can feel that as a product of respiration. When you breathe out, you get foggy glasses or moisture in your mask or whatever it is. So the end products of this are carbon dioxide, the fully oxidized carbon. The electrons have all been stripped out. Those electrons eventually got transported onto oxygen to produce water. Kind of a wonderfully elegant system, really, in many ways. So on its journey from NADH, our electron dump truck, to oxygen, it moves through 3 complexes that pump protons, 4 in complex 1, 4 in complex 3, and 2 in complex 4, for a total of 10. It's not shown on here, but FADH dumps its electrons later in the chain. And the reason for that, if we're sticking with our dump truck analogy, is the carrier, even though it still has 2 electrons, isn't as high up the hill. So we imagine a carrier like halfway up this wall, dropping its rocks through. It's not going to have as much energy to move as many protons, and so it can only move 6 protons. Okay? And as I said, the real reasons, the energetics for that we can calculate, we'll look at in other courses. And so what we end up with here is we can count up the number of protons depending on which electron carrier we're using, NADH or FADH2. So this leads us to what was called the chemiosmotic hypothesis. And this was proposed by Peter Mitchell in the early 1960s, and he won the Nobel Prize for this. And what he reasoned was that there is this process called chemiosmotic coupling. And what happens is that we have one process, this electron transport chain, that's moving protons. And that's its own contained system. So it works to push protons across a membrane into the intermembrane space. There's a second system that utilizes that proton gradient and the favorability of those protons moving back across the membrane, and that's what drives ATP synthesis. So the idea behind this chemiosmotic hypothesis was that it's two components that are actually separate. We have an electron transport chain, and we have ATP synthase. And this was demonstrated in the mid-1970s by Racker and Stakenius. And this is a really beautiful experiment that they did. So what they did was they made artificial vesicles, so with a lipid bilayer. So it's not actually a cell or a mitochondrial membrane or anything like that. They reconstituted this membrane with mitochondrial ATPase. So that's ATPase from a mitochondria. Instead of using an electron transport chain from the mitochondria to move those protons, they instead used this transporter called bacteriorhodopsin. Bacteriorhodopsin is found in it's found mainly in archaea, but some bacteria. And this is a method, it's a light-activated pump for moving protons across a membrane. So what it does is in the presence of light, it picks up protons from one side of a membrane and physically moves them to the other side. It's not from the mitochondria. So if you have both of these in your vesicle and you have no light present, nothing happens. It just sits here. So they have a bovine mitochondrial ATP synthase and this bacteriorhodopsin. However, what happened when they hit it with light was this thing managed to produce ATP. Because the bacteriorhodopsin moved the protons into, so it moves the protons into the intravesicular space, or whatever you want to call it, making a proton gradient, which then flowed down and caused the production, as we can see on the right here, of ATP. Demonstrating that the electron transport chain and the ATP synthase are separated function, and that proton gradient is what's causing the production of ATP. Questions about the experiment? Okay. So this is kind of the guts of the transporter. The transporter has a series of units. And in the mitochondria, there's 10 of these, and they're called C units. Okay, the C units. And there's this component here, this purple component. And what we can see is while the other transporters we've seen, remember, they would go all the way through. If we look at this one, actually it stalls out partway through. So this passageway doesn't traverse the entire membrane. What happens is the protons move into there, and then they bind to one of these C units, this one here. And then the whole thing makes one turn, a turn of one unit. And then the second proton comes in, and then that would bind to this unit, which will then turn, and so on. And then when they get all the way around to here, they can flow through. So this acts kind of like a turnstile. So a proton drops in to the transporter, moves into a C ring. It has to go all the way around, and then it drops through, because that completes then the channel. And so what we're doing is we're using that proton gradient to physically, mechanically turn this thing around. And we can take advantage of that, and we can use what's called a stator and rotor. So what we have here is two subunits, the F0 subunit, all of the lettering is the lettering we use, and the in the F1 unit, everything is in Greek lettering. But importantly, what we have is this C ring we know spins around, okay? And it's attached to this gamma stalk, which also spins around, okay? So this whole thing is spinning. In contrast, the A component, the B, and importantly, this alpha beta ring are fixed in place. And so what happens if you have something that's fixed in place and you put a stalk into it and you turn it? It shifts through. So this stalk, shown in green here, as it rotates, it changes the shape of those beta subunits. So let me back up here. When we look at this part of the structure, which is actually where the ATP is produced, it's composed of alpha and beta subunits. And we always just ignore the alpha because all the really good stuff happens in the beta, right? And so we can draw the beta subunits, as we see here below, kind of in three different colors. And that stalk, that gamma stalk, is stuck into the middle of them. So this thing's like a lollipop. They call it a lollipop head. But as that stalk moves, we understand that it can alter the structure of the beta subunits. Okay? So the way we have this drawn, I wish I could blow it up, but I don't think I can. The way this, if this green part of it is what it's pointing to, will be in the open conformation. So if we look all the way through here, anytime that's pointing, that's the open conformation. So as this sweeps through, and then to the right of that is in the L or loose conformation, and to the left of it is in the T tense conformation. So if we look all the way around, to the right of it, loose, left, tense, right of it, loose, left, tense. What this means is as this sweeps through, the top's turning, it's turning the stalk. As it sweeps through, those beta subunits are changing structure, okay? So they're going from the open form to the loose form to the tense form. And what happens when we change the structure of a protein? We change its function. And that's exactly what we have. So when it's in the O state or open state, ADP and orthophosphate come in. In the loose state, which will be the next, so if we just keep our eyes on the yellow one, first, let me erase this. First, our ADP and orthophosphate come in and they're bound. Okay? Then it starts to turn. It's now in the loosely bound or loose state, and they're coming together. Now, in the next step, it shifts to become the T state, which is what encourages the formation of ATP from ADP and orthophosphate. And then what's going to happen in the next step, which we don't have drawn on here, it's going to go back to being in which form? Open. And in that time, it's going to release the ATP. And what this means is for each complete turn of the C ring at the top, for each one rotation, we get 3 ATP. One from the yellow subunit, one from the orange, and one from the red. Okay? So we can do some fancy math on that. And we know the number of C subunits. We know there's 10. So what that means is for every 10 protons going through, that C ring goes all the way around. Okay, so for every 10, it does a full rotation. And so for every 10 protons that are moved across the membrane, Okay, we should produce 3 ATP. All right. And so that means that each ATP requires 3.333333333333 protons. Okay? It turns out that's not quite correct because some of those protons are used for other types, they're used for other systems. So some of those, not all of the protons move through ATP synthase. Some of those protons are used for other forms of indirect transport across the membrane. So if you actually do the energetics of it, while physically 10 have to go through to make 3 ATP, that's true, but the thing is, every 10 protons you move into that intermembrane space, not all 10 of those will go through ATP synthase. Some may go through another system. And so it really turns out that it's for about every 4 protons move into the intermembrane space, you get 1 ATP, approximately, if you actually calculate it out. Experimentally, not. If you calculate it physically, you get 3.33333. But if you experimentally determine it, it's more like 4. So why do we care so much about that? Well, that tells us how much ATP we're getting from NADH and FADH2. So remember that for glucose, by the time it got to the end of the TCA cycle and had been fully oxidized, we'd only produced 4 ATP by the end of the TCA cycle, not of stellar yield. However, if we look here, each NADH moves 4, 4, 2, 10 protons, and that translates to 2.5 ATP. And for every FADH2, which moves 6, we get 1.5 ATP. Okay. And so this is why most of our energy actually comes from, most of it actually comes from the mitochondria, because that's where oxidative phosphorylation is occurring. This is just another view of it, showing physically that it moves across into the intermembrane space. Yes?
Sorry, what is oxidative phosphorylation in comparison to electron transport?
Oxidative phosphorylation is the whole process. We're oxidizing the carbon source, and we're phosphorylating ADP to ATP.
Okay.
So that's what oxidative phosphorylation is. This can be disrupted or uncoupled, the formation of the proton gradient and the ATP synthase. It can be disrupted or uncoupled, and a couple things that may disrupt it are different poisons. So things like cyanide, okay, inhibits the electron transport chain. We can also get uncoupling by what's called DMP here, dinitrophenol. Dinitrophenol, the way this toxin works is it picks up the protons in the intermembrane space. Okay, so it picks up the protons and it moves them back into the matrix. And in this case, it does that without moving them through ATP synthase. And so the way these poisons and uncouplers work is they either stop the electron transport chain or they prevent ATP synthase from being used. And these, of course, can kill you because your body now will stop producing enough energy. Okay? Because we're dependent upon that high yield that we get out of the mitochondria. There's another way that we get uncoupling, and this can be beneficial. If we look at infants, their organs are surrounded by something called brown fat. Who's heard of brown fat before? And what's the role of brown fat? Do we know? Does anyone remember? It generates heat. So it's a method of generating heat around the vital organs. And the way it actually works is it utilizes this complex called thermogenin, which is a transporter in the inner membrane of the mitochondria. So this takes the proton gradient, and as the protons move through here, what it does is instead of generating ATP, it physically generates heat. So remember, we can convert energy into different forms. In the case of producing ATP, you've converted that movement through the transporter into a chemical bond, the ADP to the phosphate. In this case, we're taking the energy from moving the protons through to convert that to heat. Okay? This is just showing us, this will come up again later. When we move protons, for example, we create a chemical gradient because you have more protons on one side than the other, but you also produce an electrical gradient because you're moving a positive charge from one side to the other. And so that's what this is describing. We won't be looking at this with respect to the proton gradient, but we'll be looking at electrochemical gradients when we talk about signaling in the synapses, so action potentials or nerve impulses. So the top we see what we got out of glycolysis, 2 ATP, not so thrilling. From the TCA cycle, along with glycolysis, we get 4 ATP. But if we look at this using oxidative phosphorylation, it's approximately 30 ATP, maybe as high as 36. It fluctuates a little bit based on a number of factors, but we can anticipate we would get at least 30 ATP out of it. And this is taken from, I think this is from no, this is from this textbook. And this just shows us kind of the breakdown of where the ATP comes from. So a good study guide for you. And that's kind of mitochondria in a nutshell, really. Are there any questions? So hopefully we've done it at a level that allows you to understand it without being sort of overwhelming. And again, we will spend more time on this in 299, and we'll look at really how this works, the energetics behind it. We're going to move now to another ancient trapped bacterium, and we're going to look at chloroplasts. So here we have our proto-eukaryote that has its nuclear embedded or encased genetic material. It's picked up, in this case, they're saying it's from an ancient purple bacterium. More often you hear that the mitochondria is actually the result of a different type of bacterium. And then what they've done in the bottom is they've picked up an ancient photosynthetic bacterium, which is a type of cyanobacteria. Cyanobacteria are also known as the blue-green algae, and we're familiar with those living in Victoria because we get these algal blooms, especially at Elk Beaver Lake every year. And they can be toxic. They're especially toxic to dogs. So the process of photosynthesis here is the same as in chloroplasts. And the same holds true in that the chloroplasts have DNA. They carry out replication, transcription, translation that are very similar to what we see in the cyanobacteria. And the structure of them has similarities to the cyanobacteria. Generally, what we're looking at here is we're getting energy from light, we're getting carbon from carbon dioxide, we're inputting water, and we're producing C6H12O6, which looks like a carbohydrate. And that's exactly what we're producing. So we're producing a sugar. So the term photo, the energy is from light, and synthesis because it's producing carbohydrates from carbon dioxide, which we sometimes also call carbon dioxide fixation. Chloroplasts are actually super cool when we look at them because they have all these neat internal membranes. So there's an outer membrane and an intermembrane. And in this case, that intermembrane space is not of any interest to us. Okay, so we're not going to, the protons don't move into the intermembrane space. The interior, what would have been the cytosol of that ancient bacterium, we call the stroma. And within the stroma, there are these stacked membranes. And if you were to cut inside those membranes, we would see they're hollow, and there's like what's called a lumen, a thylakoid lumen in there. So we can think of these as being like a stack of Smarties, and the delicious chocolatey core is the thylakoid lumen. And the outer shell is a single lipid bilayer. And so the bacterium itself actually has these internal membranes as well. And that's an example of bacteria really having organelles, more or less. But in this case, we have this thylakoid lumen, and that's the one that we're kind of interested in because that thylakoid lumen is where the protons are going to go. And those thylakoid membranes are where the photosynthetic machinery is. And so what happens is that when we get activation of light, protons get pumped into the thylakoid lumen, and then, of course, there's a concentration gradient. The protons flow down, back into the stroma through ATP synthase, and we get production then of ATP. Yes?
Sorry, which wavelengths of light?
We'll talk about that in a minute, so yeah. Also, inside, we can see that carbon dioxide gets fixed, and we produce carbohydrates. Okay? So sometimes these are called the light and the dark reactions. The top reaction, where we make the ATP, are the light reactions. And the dark reactions, we actually utilize that ATP that we produce. It's a redox reaction, and in this case, the little dump trucks, the electron carriers we're filling are NADP+, which when it picks up electrons becomes NADPH. So those electrons that we harvest during the photo part of photosynthesis and the ATP are used in connection with carbon dioxide to produce sugars. So the way this works is that there are these photosystems. Photosystems have a series of pigments that collect they collect specific wavelengths of light. Okay? And depending on the actual organism, there can be differences in what pigments they absorb. So for example, there's things called purple bacteria that absorb the light that leaves the purple behind. Here, what we end up leaving behind is the green, and so that's why the green is what we see. So they harvest light, and they basically take the energy of it and they bounce it into this reaction center, and the reaction center is where all the fun happens. Why are we doing this? So we know that we're going to try to produce a proton gradient. Okay? It turns out that this is just another redox reaction. And if we look at the left for our mitochondria, okay, so if you imagine there's kind of a line partway down the roof, if we imagine that's on the hill where our NADH is, that was going to dump our electrons down through the electron transport chain, our NADH got its electrons initially from different organic compounds like glucose, and they were actually farther up. So they could directly drop their electrons onto NAD+. When we're looking at photosynthesis in plants, those electrons, which eventually we're going to send through our electron transport chain, the electrons we harvest are actually already at the bottom. And we harvest those electrons from a molecule called water. And if we're pulling those electrons out of water, we can't put them into NADH or NADPH. So what we do is we use the energy of light to pull them up and dump them in. All right? So we use light energy to pick them up and dump them into the NADPH. Well, really what we're dumping them into here is the electron transport chain initially, and then they'll flow downwards. What happens when we take the electrons out of water? What do we end up with as a waste product? Oxygen, exactly. Okay? So that's what we see here. What we're going to do is we're going to get those electrons from water, we're going to use light energy, and we're going to move it directly through an electron transport chain. So we don't even need to use NADH here. We just throw those electrons up the hill, and they'll flow down, pumping protons. Okay? What we'll see, however, is that we need some NADPH because we need those electrons to make sugars later, and we'll look at how we deal with that in this slide. So this is looking actually at the energetics of it, and so this is our hill that we've always been talking about, and electrons can flow down this. So we start with our water, and the electrons go into the photosystem P680. This absorbs light at 680, excites it, and then it moves down, down, down to the second photosystem, which is actually called photosystem 1. So the first photosystem is photosystem 2, and that's used solely for harvesting electrons. So its purpose is to harvest electrons from water. All the fun stuff happens in the other photosystem, so that's why it's called photosystem 1. And we can see as it traveled on its journey, it moved through this cytochrome B6F that pumps protons into the thylakoid lumen. Okay? And so what this can do is we can now excite this, and I'm going to move to the next slide. Because once an electron is in photosystem 1, we can just keep dumping it down the hill, moving those protons, and then re-exciting it. And that can just continue forever and ever and ever and ever and ever and ever until our cell dies. Okay, people good with that concept? So once we get into photosystem 1, we can use this to just keep pumping protons. Throw it up the hill, pump the protons, throw it back up the hill, protons, and every time you do that, you need to use light, right? To throw them up the hill. However, we still need to produce that NADPH. Remember, we need that to make our carbohydrate. So how do we do that? Well, as it's flowing down the hill, we can just bleed it off. So it's going down the hill. Instead of returning this way, what happens is we divert it. What we do instead is we divert it so that it flows down into NADP plus to make NADPH. Okay? So this is how we trick it into filling the electron carriers. That electron, however, is now lost. So what do we have to do? Go back to photosystem 2 and harvest another electron from water. So in the first photosystem 1, we either use them to produce protons or to fill up electron carriers. And when we fill up electron carriers, we call that reducing power. And so you'll often see in textbooks, they'll say that to make carbohydrates, we require carbon dioxide, ATP, and reducing power. And so that's what that means. We need electrons. And that's really all there is to that first part, the light reactions. It's just a modified electron transport chain. that utilizes light to energize it. Any questions? Okay. So if we compare the mitochondria and the chloroplasts, we can see there's actually a lot of similarities. We're creating a proton gradient across the membrane in an ancient trapped bacterium. That proton gradient is then used through a specialized transporter called ATP synthase to produce ATP from ADP and orthophosphate. Orthophosphate is inorganic phosphate. Phosphate that's just floating around. Okay. Here we see it in more detail. This is more detail than you need to know it in, but if it's something that you're really interested in, here's the figure, and you can follow through in the textbook. It's got more complicated descriptions, but we only need to know it for the final exam at the level of detail that we covered in class. Let's go to the dark reactions, fixing the carbon dioxide. So this cycle was worked out by Calvin, Benson, and Basham. It's sometimes called the Calvin-Benson cycle, but mostly now it's always referred to as the Calvin cycle. It's not clear to me why, but only Calvin got the Nobel Prize for this work in 1961. Most organisms that utilize carbon dioxide as a carbon source, and they fix it into carbohydrates, most of them use this cycle, but it's not the only way to fix carbon dioxide. It's just the most common, and this is the only way that plants do it, so plants and algae. Again, we're going to try to simplify the cycle a little bit compared to other years I've taught this, but it's still a little bit complicated. It involves a lot of moving carbons around, and this is kind of a nice way of showing it. Each of those gray spheres, of course, is a carbon. And where we want to start is we want to start here with 3 5-carbon molecules. We take 3 5-carbon molecules and we add 1 carbon dioxide to each of them. So now we have 3 6-carbon molecules, okay? So we had 3 5-carbons, so 15 carbons total. Now we have 3 6-carbons for 18 carbons total. And those 3 additional carbons came from 3 carbon dioxides. What we do is we then put in ATP, NADPH, so electrons, and what we end up is we end up producing 6 of this molecule, which is called glyceraldehyde 3-phosphate. And it turns out this glyceraldehyde 3-phosphate, if I go all the way back in the most irritating way possible, you're welcome. Actually, we can see that that molecule, glyceraldehyde 3-phosphate, is an intermediate in glucose, glycolysis. And the cool thing about glyceraldehyde 3-phosphate is it can either keep being broken down, or we can reverse that process in a process called gluconeogenesis to produce glucose. And that is what we do. So we go through here, we produce 3 glyceraldehyde 3-phosphates. 1 of them we bleed off. Why do we only bleed 1 of them off? Well, we need to keep the Calvin cycle going. So we can't just utilize these 6 glyceraldehyde 3-phosphates. So now this one's gone, and this leaves us with 5 3-carbon molecules that are shuffled and reassorted back to our 3 5s. Okay? So this is 15 carbons. This is 15 carbons. 3 have left. Those were the 3 that came in through carbon dioxide. And these then, if we go back to this slide, we can see that those can go on to be converted into glucose, which can then be converted into starch. And so that is everything you need to know about organelles. Okay? So we'll leave it there, and Friday, no class, your exam. We'll meet up again on Tuesday.